IB Bio Genetics Part 2, after a short introduction where I define some terms, will have its focus on one pattern of heredity, known as dominant recessive. As well, I will review the techniques involved in solving genetics problems. The essential idea is the inheritance of genes follows patterns. Here is the outline of genetics movies available for your study. Use this outline to find the movie you need. This movie is focused here. Genetics is the study of heredity, and to that extent it is the study of the patterns in the way alleles are passed from one generation to the next, and the way in which alleles express themselves in individuals, families, and populations. Here are the first 3 IB syllabus statements of importance. The 2 alleles of each gene separate into different haploid daughter nuclei during meiosis. State that gametes are haploid, so contain only one allele of each gene. State that fusion of gametes results in diploid zygotes, with 2 alleles of each gene that may be the same allele or different alleles. Individuals inherit alleles, one from the father and one from the mother. Gametes carry one chromosome of a homologous pair due to meiosis. Thus, with only one chromosome of a homologous pair, gametes only carry one allele of each gene. Gametes fuse to restore the diploid condition. Diploid nuclei have 2 alleles for each gene because they have pairs of homologous chromosomes. With this slide, I will start using letters to represent alleles. A capital letter represents one allele, while the lowercase letter represents a different allele for the same gene. We inherit one allele, such as capital A, from our father, and we inherit one allele, such as lowercase a, from our mother, and that would produce the diploid condition of capital A, lowercase a. This is called the genotype. This is the diploid condition because we have the 2 alleles together. The homologous pairs are together. When the gametes fuse, the diploid condition is restored, and we could have capital A, lowercase a, or 2 alleles of the capital A type, or 2 alleles of the lowercase type. We will provide some terms later on in this movie. The genotype is the specific composition of the alleles for a gene in an organism. The genotype determines the phenotype. The interaction of the 2 alleles results in the observable, the visible condition called the phenotype. Mendel is often credited as the father of genetics for his work in understanding the heredity patterns of certain chosen characteristics in peas. He knew nothing of DNA, chromosomes, or meiosis, but the theories he put forward, based on the data he collected, were correct. Mendel grew a lot of pea plants, and it was the magnitude of his data collection that has earned Mendel such respect as a scientist. Once he had counted the offspring from a cross, he calculated ratios and developed his theories. To perform a cross, Mendel took the pollen, sperm cells, from a pure breeding line of plants, say the white flower plant, and brushed it near the egg of another pure breeding line of plants, in this case, the purple flower plants. So when he crossed purple flower plants with white flower plants in this way, he collected the offspring, the seeds, and he grew the seeds to see what the flower color was in the offspring, and he noticed all purple. First, he realized that the hereditary particles, as he called them, remember, he knew nothing of DNA, genes, or chromosomes, did not simply blend. In other words, if you cross purple and white flowering plants, would the resulting plants be some intermediate color? No, blending did not happen. The purple color seen in the offspring was purple. The hereditary particles were discrete structures, but he also wondered why all the offspring were of the purple phenotype. Where did the white phenotype go? So here's another way to look at what he did. He started with pure breeding parents, and I'm emphasizing this because, as you will see, this is important information for understanding the cross of these 2 parent types. Be patient. Starting with pure breeding purple and white parents, the cross produced offspring, all of which had the purple flower. The offspring are called the F1 generation. All the offspring were purple, no blending, but where did the white phenotype go? So then, Mendel took the purple flower offspring and mated them among themselves. Take note, if you're thinking carefully using what you already know about genetics, the purple flower parent does not have the same genotype as the purple flower offspring. You can see here the F1 first generation are called hybrids. This is a hint. So, take note, he crossed the F1 purple flower plants with other F1 purple flower plants to produce the F2, the second generation. And look what occurred. The white phenotype reappeared, and his numbers, big numbers, resulted in an approximate whole number ratio of 3:1. Notice, by the way, that his numbers were not exactly 3:1, but they were very close. Mendel followed many different characteristics, and he counted big numbers. And he found a pattern in the phenotypic ratio of the F2, the second generation, always close to 3:1. From this data, he was able to theorize about what was going on, and I will follow up on what he thought was going on after I explained what happened in his crosses. I want to note that the characteristics Mendel followed displayed the dominant recessive heredity pattern, which is the focus of this movie. So with no further ado, let's look at the dominant recessive heredity pattern, the subject of genetics movie part 2. With this slide, you can see the other patterns of heredity that I will cover in the subsequent parts in the genetics series. And here are two relevant IB syllabus statements to find the dominant allele and the recessive allele using appropriate examples. Define the following terms: homozygous and heterozygous. The dominant allele is always expressed when it is present. If the genotype is homozygous A, the dominant allele is expressed in the phenotype. In this example, the phenotype produced by the dominant allele is curly, let's say curly wing. If the genotype is heterozygous A, the dominant allele is still expressed and the phenotype of the individual is curly. Maybe you can define homozygous and heterozygous now. Homozygous refers to the genetic condition when both alleles are the same in the genotype. Heterozygous refers to the genetic condition when the two alleles in the genotype are different. The recessive allele is not expressed in the presence of the dominant allele. The recessive allele is masked or hidden in the presence of the dominant allele. The recessive allele is expressed only when both alleles in the individual are recessive. In other words, homozygous recessive. The phenotype would be straight wing. So with alleles in mind and the locus for flower color located here on the chromosome, let's take a different look at what Mendel did. He crossed pure breeding purple flower plants, in other words, homozygous purple, with pure breeding white flower plants, homozygous white. So here's the cross, pure breeding purple flower plants crossed with pure breeding white flower plants. We can use a Punnett square to show the separation of alleles due to meiosis, the white flower haploid gametes here and here, with the purple flower haploid gametes here and here. Single letter indicates haploid. Fertilization brings the two alleles back together in the formation of diploid offspring. Close examination of these individuals provides the expected ratios of the offspring. Notice all the F1 offspring were of purple flower phenotype. The phenotypic ratio was 4 purple, 0 white. The genotypic ratio was 4 heterozygous purple flower, 0 homozygous purple flower, and 0 homozygous white. As a result of noting that all the F1s had a purple flower phenotype, Mendel was able to deduce that the allele for purple flower was dominant to the allele for white flower. Capital, lowercase symbols are used to denote dominant and recessive, respectively. I've stayed with this slide for a moment. You might remember that I mentioned that the genotype of the purple flower parent was different from the purple flower F1. Take a look. And you might remember that crossing pure breeding purple with pure breeding white did not result in an intermediate color. The alleles are discrete. They do not simply blend as a blender might blend colors. You now know the dominant allele is always expressed when present. You might also remember that Mendel wondered why no F1 plants were white. Maybe you can see what happened next. So here is the cross among F1 plants, F1 purple flower plant crossed with an F1 purple flower plant. A cross like this, by the way, involving two heterozygous individuals is known as a monohybrid cross. We're following one gene, the flower color gene in this case, thus mono, and the individuals being crossed are both heterozygous, a hybrid of the dominant and recessive alleles, thus monohybrid. Meiosis separates the alleles here and here. This happens during metaphase, anaphase 1 of meiosis. It's called segregation. Random fertilization restores the diploid condition, bringing alleles together again, resulting in a phenotypic ratio of 3 purple to 1 white. 3 purple to 1 white. The genotypic ratio is 1 homozygous dominant, 2 heterozygotes, and 1 homozygous recessive. 1 homozygous dominant, 2 heterozygotes, and 1 homozygous recessive. Now these are expected ratios, and when counting big numbers, the actual ratios will be close to, but not exactly, the expected ratios. By way of example, if fertilization is truly random and lots of offspring are counted, the probability is increased that the phenotypic ratio will be close to 3 to 1, albeit never exactly 3 to 1. Now here's a slide to emphasize the difference between the F2 genotypic and phenotypic ratios resulting from a monohybrid cross. Monohybrid cross results in phenotypic ratio of 3 to 1 and a genotypic ratio of 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. Study this slide to bring these ratios under control. This IB syllabus statement should feel familiar. Determine the genotypes and phenotypes of the offspring of a monohybrid cross using a Punnett grid. Mendel put forth these theories. As he worked in the late 1800s, he was never able to verify them, but we know today that he was correct. Alleles account for variations in inherited characters. For each variation in character, an organism inherits 2 alleles, one from each parent. If the 2 alleles in an individual differ, then the dominant allele is fully expressed in the phenotype. The recessive allele has no effect on the phenotype. The 2 alleles segregate during gamete production. Again, notice Mendel's F2 phenotypic ratios, starting with crosses of pure breeding lines of plants. All of them were close to 3 to 1. Also notice he happened to choose, maybe he was lucky, a lot of characteristics that displayed the dominant recessive heredity pattern. If he had chosen a characteristic controlled by a different heredity pattern, he might have been confused and not developed the theories that he did. Maybe he did bump into such a characteristic, but chose to ignore it. Let me emphasize that using a Punnett grid, as we did in the examples that preceded this slide, we produce expected ratios, but that actual numbers, actual ratios, are never exactly the expected ratio, and this is due to random fertilization. With the remainder of this movie, I want to strengthen some of the concepts that I've presented so far and show you some different ways of solving genetics problems as they present themselves. I've spoken about pure breeding parents, and I've mentioned that a pure breeding individual is homozygous with respect to the gene in question. A pure breeding line, seen in this slide, is one where you cross one individual with another and the offspring have exactly the same genotypes as the parents. You can see that here. Also, some people prefer this spider diagram to determine the outcome of a genetic cross rather than a Punnett grid. Meiosis separates the alleles, and then random mating produces expected ratios. In this pure breeding case, all the offspring have exactly the same genotype and phenotype as the parents. The IB syllabus specifies the Punnett grid. so I won't show you a spider diagram beyond this slide and the next. This cross, in spider diagram form, is not a pure breeding cross. It's a monohybrid cross. Each parent is heterozygous for a single gene. I'd like you to understand the terminology here. Monohybrid, single gene, heterozygous, both parents. Meiosis results in the segregation of alleles, and random mating results in the 3:1 phenotypic ratio because this is a dominant recessive heredity pattern. The genotypic ratio is 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. Here is the monohybrid cross using a Punnett grid. Can you write the phenotypic ratio and the genotypic ratio that results from this cross? Punnett squares display Mendel's law of segregation. The segregation of alleles during meiosis creates genetically different gametes. Then during sex, gametes fertilize to produce the diploid offspring. Mendel's law of segregation is every individual contains a pair of alleles for each particular characteristic, a pair, which segregate or separate during meiotic cell division into haploid gametes. So I'm back to showing you homologous pairs and a single gene cross. We have an individual who is homozygous dominant for the characteristic in question, crossed with an individual who is heterozygous for the gene in question. These two letters represent two alleles on a homologous pair of chromosomes, and these two letters represent, again, two alleles on a homologous pair of chromosomes. Remember that the alleles separate during meiosis 1. A carrier is an individual who carries the recessive allele. The recessive allele is masked by the presence of the dominant allele. The heterozygous individual in a dominant recessive heredity pattern does not express the recessive allele, but could pass the allele to their children. Here's the same cross that I showed in the last slide, but in this slide I've showed you the expected outcome for the offspring using a Punnett square. You can see that 2 of the children from the cross are also carriers. Once again, notice the difference between the phenotypic ratio and the genotypic ratio. Let's solve a problem, something simple, using a Punnett square. In pea flowers, purple flower color is known to be dominant to white flower color. What is the outcome if a heterozygous purple flowered plant is crossed with a white flowered plant? Meiosis puts the alleles for the purple flowered plant here, and the alleles for the white flowered plant here and here. Random mating brings the alleles back together into the diploid condition, and you can see the results as they unfold here. We're going to move to the next slide to see the phenotypic and genotypic ratios. Here are the phenotypic and genotypic ratios. I'll let you spend a bit of time with this slide to ensure that you understand how it is this phenotypic ratio emerged from this cross and this genotypic ratio emerged from this cross. Let's solve a problem backwards, working to get at the question that's been asked. In pea flowers, purple flower is known to be dominant to white flower. The offspring of a cross are examined for flower color. Out of 240 total plants, 62 are white and 178 are purple. What are the genotypes of the parents? This is the question. What are the genotypes of the parents? Now we can see that the phenotypic ratio of 178 to 62 is close to 3:1. Thus you might already know the genotypes of the parents, but let's keep going using the available information. Since there are white offspring, we know that each parent must have a white allele in their genotype, and we can place that allele on the Punnett grid here and here, and we can put a white offspring here. Now, since purple flower outnumbers white 3:1, we can put a purple allele here and here and here, and thus with that in place we can get at the alleles. We would have a purple allele here, a purple allele here. With that, we can see that one parent is heterozygous purple and the second parent is also heterozygous purple. We can fill in the Punnett square with all the information that we have in front of us. Here's what the cross looks like. Remember, these provide the expected ratio. This is the actual ratio. We know that both parents are heterozygous in order to produce this particular outcome. Let's solve another problem backwards. In pea flowers, purple flower is known to be dominant to white flower. The offspring of a cross are examined for flower color. Out of 240 plants, 124 are white and 116 are purple. What are the genotypes of the parents? This is the question. What are the genotypes of the parents? We can see the approximate 1-to-1 ratio among the offspring. Based on this, you may already know the genotypes of the parents, but let's continue. Now, there are white offspring, so both parents must have the recessive white allele. There are also purple offspring, so one of the parents must carry the purple allele. And with that, we could take the alleles from this individual and place them right here on the Punnett grid. As well, we can take the allele here and place it on the Punnett grid. This individual will then be white. This individual will be purple. The second allele is coming. This individual is heterozygous purple. Now we can see, based on the 1-to-1 ratio, that half of the offspring are going to be white. So let's put a second white offspring here. We can see at this point half the offspring are white, and with that we know the second allele here. Now we have all the alleles in place. In fact, the second adult is homozygous recessive, and we can see this individual heterozygous with its alleles here. This individual is homozygous recessive, its alleles here. Let's look at the next slide. Here are the genotypes of the parents. Here are the offspring. These are expected ratios. This is the actual ratio. It approximates 1 purple to 1 white, which is exactly the information given in the question. Here is a problem to solve that follows an autosomal recessive allele that causes cystic fibrosis in the homozygous condition. Cystic fibrosis is an inherited disease caused by a recessive allele on an autosome. Mary and Harold do not suffer from cystic fibrosis, but both of their mothers did. What is the probability that they have a child with cystic fibrosis? I've given information here that cystic fibrosis is caused by a recessive allele, so I've designated the recessive allele F for fibrosis and the dominant allele F for no fibrosis. We know that both Mary and Harold do not suffer from cystic fibrosis, so Mary and Harold each have a dominant allele. We also know that both of their mothers had cystic fibrosis, which means that the mother was homozygous recessive. That automatically means that both Mary and Harold are heterozygous. We can put their alleles in here following meiosis. We have gametes with single alleles. We can go through random fertilization to produce the expected ratio of offspring, and we would expect in this case that 1/4 of their offspring would have cystic fibrosis. So the probability that they have a child with cystic fibrosis is 1/4 or 25%. Here is another question on cystic fibrosis, which is an autosomal recessive allele. Using these symbols for the normal and recessive alleles, give the genotypes of Jane, Daniel, and their children. Jane and Daniel are not sufferers of cystic fibrosis, but you can see that they have a child who suffers from the disease. Their child must be homozygous recessive for cystic fibrosis and must have received an allele from each Jane and Daniel. But as Jane and Daniel are not sufferers, Jane and Daniel are both heterozygous. We can answer the second question here. If Jane and Daniel have another child, what is the probability the child is healthy, a sufferer of fibrosis, or a carrier of the allele, in other words, heterozygous? We can predict that Jane and Daniel would have 3 out of 4 children who would be healthy. So the probability of a healthy child would be 3/4. The probability of a sufferer of fibrosis would be 1/4. And the probability of a carrier, a heterozygote child, not a sufferer, would be half at 50%. As a last note, this individual, the other child of Jane and Daniel, does not have cystic fibrosis, but we do not know the second allele. Here are two relevant IB syllabus statements. State that many genetic diseases in humans are due to recessive alleles of autosomal genes, although some genetic diseases are due to dominant or codominant alleles. State that many genetic diseases have been identified in humans, but most are very rare. So let's look at disease that results from dominant alleles. You might be surprised that some dominant alleles cause disease, but it's important that you do not assume dominant alleles are always beneficial or in high frequency in the population. Too often, young students of biology want to think of dominant alleles as always beneficial or always higher in frequency. The frequency of alleles in the population is determined by natural selection, not whether it's dominant or recessive. Huntington's chorea is a disease caused by a rare dominant allele. Having the allele is lethal. Disease onset begins in your 40s. Within 10 years or so, death follows. As a dominant allele, a sufferer of Huntington's must have had one parent who was also a sufferer. The allele is rare, which means that a Huntington sufferer is most likely to have only one allele. It would be highly unlikely for someone with Huntington's to have had two parents with Huntington's. The allele is rare. Thus, if a young person with the dominant allele marries someone, it is highly unlikely that the spouse will have the allele. Here's the cross, and here's the Punnett grid. These parents would expect half of their children to suffer from Huntington's. Interestingly, Huntington's chorea is caused by a lethal dominant allele that does not strike until one is older. Unfortunately, that means someone with Huntington's passes it on to their children before knowing that they have it. As I mentioned, it's easy to fall into the trap of thinking that dominant alleles are better or beneficial relative to recessive alleles. Let this bias go. Also, the dominant allele isn't necessarily the most numerous or frequent in the population. The frequency of an allele in the population is determined by natural selection, not a Punnett square. So I have three last thoughts in bringing this movie to a close, and the first is some perspective on dominant alleles. Dominant alleles are not always beneficial. I've just been through Huntington's disease as an example. Keep this in mind. And dominant alleles are not always the most common allele in the population. Remember, the frequency of alleles in a population is dependent on selection, the advantages or disadvantages of alleles in a local environment. And secondly, offspring ratios are never exact. Think carefully about offspring numbers. Look for closeness to whole number ratios. Punnett squares can deceive young students of biology into imagining that hereditary patterns appear in whole number ratios. Whole number ratios represent expected values, while actual values are dependent on the random events during fertilization. And lastly, I'd like to introduce you to the test cross. The test cross is used to determine the genotype when the genotype is not fully known. I'll show you about this in just a second. Ratios are close to expected values, but never exact. They're never 3.0 to 1. It's more like 3.2 to 1, and you have to recognize this particular ratio as 3 to 1, which would be typical of the heterozygous or monohybrid cross. So if you're looking at a cross of a heterozygous individual with the homozygous recessive, and out of 240 plants, 110 are white and 130 are purple, you need to recognize this ratio as a 1-to-1 ratio, even though the actual ratio might be 1.2 purple to 1 white. The test cross is used to determine an unknown allele when an individual has the dominant phenotype. A test cross is when the individual with the dominant phenotype but unknown genotype is mated with a homozygous recessive individual. This is the test cross. So we take the individual whose second allele is not known, we mate it with the homozygous recessive, and we take a look at the offspring. If all the offspring have the dominant phenotype, this parent is going to be homozygous dominant. But if at least one offspring has the recessive phenotype, only one is required, this parent is heterozygous. Here's the IB syllabus statement. You need to know what a test cross is. And that brings us to the end of IB Biogenetics Part 2.